Hello, dear students. We are going to study different ways of carbon fixation in plants. As we have studied, plants are C3 plants and they are C4 plants. Plants can fix carbon during dark reaction during carboxylation phase in a C3 way, and they can fix them another alternative method of carbon fixation, which is called C4 methods. And what actually these methods are, I'm going to detail them today. Starting with the very first method, that's a common method that's called C3 plants. C3 plants are those plants that are living in a normal environment and they have a normal temperature and normal air effect, and therefore they are having a normal mechanism of closing and opening of stomata, and therefore there is no effect such as photorespiration. Therefore, these plants can easily open their stomata during day and they can fix carbon dioxide and this leaf. For example, this is structure of leaf, this is epidermis, and this is these are gall cells that are making this pore that's called stomata, and this is a packed region of a palisade region, and then this is a loose region that is to be spongy mesophyll. And the mesophyll cells, the cells of a spongy mesophyll cells are site where the process of photosynthesis is taking place. At this place, in these cells, we have just specified a one single large cell. And this is to be considered as a mesophyll cell. What is going to happen during C3 mechanism? During daytime, when light is present and thylakoid in these cells, thylakoids are producing ATP and NADPH. In presence of this chemical energy, fixation of carbon dioxide is gonna happen, and in that process, plant will fix carbon dioxide and will synthesize their glucose. What will actually happen? There is a higher concentration of a carbon dioxide here in a mesophyll region. As the stomata open and carbon dioxide can come in and oxygen coming out of photosynthesis can go out. Therefore, there is no problem in performing and fixing carbon dioxide in these C3 plants. They have a higher concentration of carbon dioxide in this region, in their mesophyll region. They can take that carbon dioxide that combines with the RUBP and that can synthesize hexose, and that breaks into two triose. So the product that's coming out, a stable product that's coming out of hexose or glucose, it is 3 carbon containing. So they synthesize 3 phosphoglycerate, 2 molecules of 3 phosphoglycerate. Actually, each 3 phosphoglycerate is 3 carbon containing molecule, and when a plant during carboxylation phase, during fixation of a carbon dioxide, if produces a stable product that contains 3 carbons, we consider these plants are C3 plants. Very simple. They simply have their stomata open during daytime. They can fix carbon dioxide, and carbon dioxide is fixed into RUBP, synthesize hexose that breaks into triose. They contain 3 carbon. Therefore, these plants are termed as a C3 plants. And by this process, oxygen is coming out as well, and that oxygen will again come into this mesophyll region and that diffuses out. So they do not have a difference in the concentration of carbon dioxide and oxygen. Therefore, they do not face any kind of problem in performing their photosynthesis. Therefore, they are normal plants. They can synthesize their triose, and to these plants we consider these plants are said to be C3 plants. Easy and very simple. So this is the process of a plant. Subscribe my channel and share it with your friends, and come in also. Let's learn together.